quick show of hands. Not that I can see you, but how many of you have ever written a line of code? Python, C++, maybe even just some HTML. Now, what if I told you that right now, in labs around the world, scientists are treating DNA, the literal blueprint of life, as an operating system? And they're debugging it, just like software. The question is, would you trust yourself to rewrite the code of life? Here's how the analogy works. Think of a cell as a computer. The hardware is the cellular machinery, ribosomes, membranes, mitochondria. The operating system is the natural genetic code. And diseases like cancer or sickle cell anemia, those are bugs in the system. For decades, scientists were just observers. They read DNA. They watched what happened. Then they started editing, like using a word processor to fix a typo. Cut, paste, replace. Simple. But then something shifted. Scientists asked, what if we could compile new DNA from scratch? What if we could write a piece of code that makes a bacteria glow green when it detects arsenic in your drinking water? And what if we could upload that code like a software update? Proto, the programming language. So how do you actually write biological software? It's not like typing in Python. You have 4 letters: adenine, thymine, cytosine, guanine. That's it. 4 chemical bases. The entire code of life. But here's the game changer. In June 2026, Stanford University and the Arc Institute released Proto, a high-level programming language specifically designed for generative biology. Proto works on 4 core concepts called primitives. Tell it you want to design a promoter, the piece of DNA that turns genes on and off, and it will generate thousands of candidates, test them against constraints, and optimize the best ones. It's like having ChatGPT for DNA. In human cell tests, Proto designed introns, DNA sequences that are spliced out during protein production, with 32% achieving significant cell type specific splicing. By contrast, a similar earlier study saw less than 7% success after testing over 100,000 sequences. Proto achieved better results with far fewer candidates. For promoter repressor design in bacteria, 45 out of 86 pairs produced gene expression more than 10 times higher than controls without a promoter. Proto is currently available as a preprint on bioRxiv and has been released as an open platform, with the full software infrastructure and user interface available to the wider research community. Sin 57, rewriting the dictionary. Now think about this. What if you could completely rewrite the genetic code of a living organism? In January 2026, Cambridge researchers published a landmark achievement. They built Sin 57, an E. coli bacteria with a completely rewritten genome. Natural genetic code has 64 codons, 3-letter words that tell cells which amino acids to make. Sin 57 uses only 57. The team made over 101,000 changes to its DNA over 4 years of work. Here's why. By compressing the genetic code, they freed up space. Now Sin 57 can be programmed with new functions that natural bacteria can't perform. It grows 4 times slower than normal E. coli. That's the trade-off. But it has a superpower: viral resistance. Viruses can't hijack a code they don't understand. Here's your challenge. If you could give Sin 57 one new function, just one, what would it be? 1. Produce a vaccine on demand. 2. Eat plastic pollution. 3. Glow red when it detects cancer markers. Think about it, because here's where things get really interesting. The SpudCell, artificial life created. Scientists make living cell. You've probably seen these headlines. On July 2nd, 2026, Researchers at the University of Minnesota announced SpudCell, a synthetic cell built entirely from non-living chemical components that could feed, grow, and divide. The media went wild, but here's what the headlines didn't tell you. The SpudCell borrows ribosomes from E. coli. Without them, it can't build its own proteins. It's like having a car that runs, as long as someone keeps pushing it and refueling it every 5 miles. After just 5 to 10 generations, it breaks. The ribosomes dilute, the genome degrades, and the cells stop dividing. Victor De Lorenzo, a top synthetic biologist at Spain's National Biotechnology Center, called the hype completely hyperinflated. At no point is life created de novo. What is demonstrated is the ability to manually assemble, using pre-existing biochemical components, a cell-like system that replicates some of its functions. There's another important detail. The research hasn't passed peer review yet. It was rejected by a major journal and posted directly to the researcher's website. Real talk is, should we be worried about this technology? 1. Yes. We're playing with God's creations and it's dangerous. Or 2, no, it's just advanced engineering, like making a better car. Scientists aren't creating life. They're rearranging existing building blocks. It's like being able to rearrange Legos to make something new, but the pieces are still Legos. Real progress. The SC 2.0 project. While SpudCell grabbed headlines, another project made bigger progress. The SC 2.0 project, over a decade in the making, is on track to create the first fully synthetic eukaryotic genome. Yeast has a nucleus, like human cells. This is far more complex than bacteria. All 16 chromosomes of yeast have been synthesized, with the project nearing completion. In April 2026, researchers published a breakthrough in Nature Communications. They'd built a neochromosome 3 that relocates essential genes and stays stable for over 100 generations. And they're already planning SC 3.0, a minimal yeast genome stripped down to the essentials, aiming to identify the smallest set of genes needed to sustain life. This matters because yeast is used to produce insulin, vaccines, and industrial chemicals. A programmable yeast genome means cheaper medicines, more sustainable manufacturing, and faster pandemic response. Think about this. What would you want to see engineered with synthetic biology? Option A, self-healing buildings. Option B, personalized cancer treatments. Option C, plastic-eating enzymes. So here's the reality check. We have Proto, a programming language that actually works for biology, released June 2026. We have Syn57, a bacteria with a compressed writable genome, published January 2026 in Science. We have SC 2.0, a synthetic yeast project with all 16 chromosomes complete and a stable neochromosome verified April 2026. And we have SpudCell, a remarkable but limited cell-like system announced July 2026. We are not coding life from scratch. We are repurposing, rewriting, and reshaping what evolution already built. And honestly, that's more impressive and more useful than the hype suggests. The question isn't whether we'll program biology. We already are. The question is, how will we use this power? Let me know in the comments. Would you design something to heal, to clean, to protect? Or would you do something else entirely? Hit like and subscribe to channel for more science.